Welcome everyone. Thank you so much for being here to NUI's Ocean Series, a series of ocean-based webinars that are hosted also by the Nature Tech Collective. The Nature Tech Collective is a coalition of organizations working towards a nature-positive future. We have solution providers, startups, project developers, corporates in our community that are all gathered and collaborating towards nature positive outcomes. I'm Paige, I'm the head of community at the Nature Tech Collective. And again, this is one of the gatherings that we facilitate. So we have meetings around oceans, but also weekly community webinars or demo days. We have one at the end of June. And yeah, as everyone's kind of joining and here, I'm going to let Amal start the introduction of the actual event. Let's go.
Fantastic. Thank you, Paige. Good morning, good afternoon, good evening, everyone, wherever you're joining us from. Welcome. My name is Amal Ketata. I'm founder and CEO of NUI, Blue Finance startup, and NUI is a member of NTC. Very glad today to be presenting together with NewAtlantis Labs a really hot topic in ocean biodiversity, which is metagenomics. Today, the ocean comprises most of the living space in our biosphere, but if we had to write a catalog of what lives there, 99% of the pages would be blank. And that was a quote from Enrique Sala, who was an explorer at National Geographic. And so ocean metagenomics is emerging as a new frontier for understanding marine biodiversity, enabling more informed conservation and resource management decisions, and also unlocking economic opportunities, in particular for marine protected areas. And NewAtlantis Labs is pioneering the first marine dedicated metagenomics platform, which is transforming raw eDNA data into revenue generating products, which would benefit MPAs, but also governments and local communities, and the users, which would be the industries and investors. So I'm very pleased to welcome the NewAtlantis Labs team for a deep dive into this space. And today we have Florence Tercier, lead global development project. Florence Tercier brings hands-on experience in global conservation and innovative finance to her role as lead of global project development at NewAtlantis Labs. She has played a pivotal role in launching major initiatives such as the world's largest marine conservation debt swap in the Galapagos and the development of technology platforms for protected area management, making her a driving force for impactful cross-sector collaboration in the ocean space. Her background as program director at philanthropic foundations like Legendfly Philanthropy and Oak Foundation gives her a unique perspective on turning big ideas into action for a healthier, more sustainable blue planet. We also have Josh Espinosa, head of bioinformatics. Josh is a multidisciplinary genomics research scientist with over 12 years of experience, 30 plus peer-reviewed publications, and has led diverse research projects developing microbiome workflows, AI-based diagnostic systems, and biomarker discovery platforms. He obtained his doctorate through a joint program between J. Craig Venture Institute and Durban University of Technology in South Africa. His research expertise is in metaomics, compositional network analysis, single-cell analysis, and biomarker discovery with an emphasis in environmental and human systems. Welcome, Josh. And Jay Gutierrez, Chief Scientist at NewAtlantis Labs. He drives the convergence of marine conservation, systems biology, and AI to create quantifiable ecological assets for blue natural capital markets. He leads interdisciplinary scientific initiatives that transform complex marine biodiversity data into actionable conservation and biotechnology assets, architecting integrated analytical frameworks to connect molecular insights to ecosystem scale understanding. His expertise includes advancing multi-scale modeling applications for marine ecosystems and leading the development of knowledge graph representations of global marine metagenomics datasets and fish food webs and multidimensional query capabilities. Welcome, Josh. Florence, I'll let you take it away.
Thank you very much, Amal, Paige, and the Nature Tech Collective for inviting us today, and welcome to all the participants for joining us today. It's a pleasure to be part of this community of innovators and change makers working at the intersection of technology and nature. My journey in marine conservation has taught me that protecting our oceans isn't just a scientific or moral imperative. It's an economic challenge that demands new solutions, and we are here today to expose some of those. In this presentation, we will cover the current gaps and challenges in ocean conservation to unlock its value.
Excuse me,
Yeah.
excuse me, Florence. I can't see your presentation.
Josh, are
You see
you?
your desktop, not the presentation yet.
It's not yet up? Okay,
Now, yes.
Yeah.
thanks.
Thank you so much. Josh is actually doing it. Okay. So in today's presentation, we will cover the current gaps and challenges, present New Atlantis, and explore the ocean microbial complexity and the Metagenomics approach, the potential and the technology. We will present PISCES, which is the AI-driven marine platform that New Atlantis has developed using these approaches. And we will make a demo of microcosm analytics, which is an application of PISCES, and finally lay out the different partnership models and potential collaborations and development of future strategies. So I'm not going to tell you what are the many oceans crises and why it's important to protect fully at least 30% of the ocean to hold or reverse some of the threats, as you all know this. But let's just have a look briefly at the current gaps to achieve this. So despite incredible ecological value of MPAs, only 2.7% of the ocean is fully or highly protected out of the 8.3 on paper. This is due to a combination of political, financial, social, and other practical challenges. And some of the things that stand out in the way of achieving 30 by 30 is that MPA management are costly and that benefits from MPA are difficult to be monetized, making it hard for government stakeholders or funders to value the gains. And studies show that 70% of MPAs are chronically underfunded, also making it difficult to enforce protection but measure progress if there are. Philanthropy is important. It's an important support to the designation, often the science behind the creation of MPA and the implementation of MPAs, but it's not sustainable, and still marine conservation represents less than 1% of global philanthropic funding dedicated to ocean-related causes. At the same time, we have MPA managers and conservation practitioners often lacking access to the kind of high-resolution, actionable data that are needed to monitor biodiversity and ecosystem health. And remarkably, less than 1%, as mentioned earlier, less than 1% of marine genetic diversity has been sequenced, meaning that we are just scratching the surface of what's possible in both conservation and innovation. So this is what prompted a couple of serial entrepreneurs, Courtney Nicole Gould and her husband, Gordon Gould, to create NewAtlantis Labs in 2021, a startup that is aimed at responding to those pressing challenges. So in short, it's a for-profit company. It's dedicated to transforming marine data with the use of the most advanced genomics, AI, and open analytics. It is generating cutting-edge insights for monitoring and forecasting tools for conservation purpose, but also generating financial assets through marine biodiscovery, risk assessment modeling, and market-based instrument for valuing actions to conserve or restore marine ecosystem health, and driving financial revenue that in turn can support marine conservation through a benefit-sharing model that is compliant with the international policies and treaties covering the use and benefits of genetic resources like the Nagoya Protocol or WIPO Treaty. The tech and science team that you will see Jay and Josh, but also Christian Correa, the marine ecology and biodiversity specialist engaged with us, who is also online. The team biological data sets, and also with scientific expertise in bioinformatics, computational and system biology, protein engineering, and ecology. But now Josh, the head of bioinformatics, will show you what metagenomics means in the context of the ocean and how it can be leveraged for conservation and for generating financial value for the ocean. To you, Josh.
So microbes are the key to unlocking ocean value sustainably. And marine food webs are dependent on microbes. And as you can see on the right, we have our energy pyramid. And at the base of this energy pyramid, we have primary producers, which makes up a lot of microbes and phytoplankton. And this is essential to the food web and also recycling nutrients and driving ocean chemistry. And they support all life in the ocean. So everything you love, from jellyfish to coral reefs, all the way up to megafauna like whale sharks and blue whales, all depend on microbes at some point in their lifespan. And microbial communities are highly complex, so surveying microbial biodiversity is paramount to establishing the baseline of natural systems and natural fluctuations within the system, so we can compare changes to the system to these baselines. And this allows us to create insight into larger trends, like if we're trying to track fishery populations or predicting algal blooms or disease onset. And microbes also generate valuable natural products. So bioprospecting offers sustainable commercial value to natural systems, and natural products such as biomedical and industrial enzymes can be harnessed without disturbing the ecosystem, since we're doing all of this in silico or on the computer. And with greater biodiversity, we get greater value for natural systems, and this can be value in different ways. So greater biodiversity enhances ecosystem stability and resilience. And it also supports healthy ecosystem productivity. So think like carbon sequestration, fisheries, ecotourism. And greater biodiversity also increases the opportunity for identifying novel natural products through bioprospecting and enables more holistic and powerful predictive models. So how do we go from the environment to the cloud? Well, when we process environmental samples, we do field sampling. So we do plankton tows, water filtration, sediment grabs, and then we send these samples to the lab for genetic sequencing. So this can be either DNA or RNA sequencing. And then we transform this unstructured raw data into contextualized genetic assets using different bioinformatics workflows. And then we develop interactive reports with context from our knowledge graph. And then we interpret these reports for actionable insights or intellectual property, depending on the client. So what is metagenomics? Well, it's a study of genetic material from a community in place. And I really want to emphasize in place because it's important that we study these communities as they exist naturally. So why study genetic material in a natural setting? Well, for one, we can cross-reference it with multimodal data. So there's a lot of open source satellite data, like from NASA MODIS or from Copernicus, and they're measuring like chlorophyll A or like sea surface temperature. And there's also buoy measurements as well with like temperature and salinity. So we can cross-reference all of that with our samples. And there's also microbial dark matter. So you might imagine getting some seawater, collecting it and putting it on a petri dish or some type of culture and then sequencing that. Most organisms cannot yet be cultured in lab, and most microbes can't be cultured in lab, and this is true for the ocean as well. So characterizing this microbial dark matter helps fill in the gaps in what life is capable of and gives us a fuller picture of the ecosystem. So what's the difference between the different omics technologies? The first one is metabarcoding. This is what everyone's probably most familiar with, and this is the cheapest, and it essentially says like who is there. It's cost effective for time series and broad biodiversity surveys, and it estimates the presence, absence, or relative abundance of organisms representing a snapshot of the community. And this uses targeted sequencing instead of random selection, but it's highly dependent on the databases used, unlike shotgun metagenomics and to some degree metatranscriptomics. And this is typically referred to as eDNA or environmental DNA, but technically environmental DNA also encapsulates shotgun metagenomics as well, which is the next omics technology. It's a little more expensive and it assesses who is there and what can they do. And this offers deeper insight into the community because you can reconstruct the genomes de novo and assess their functional potential. It's called shotgun because the genetic material is fragmented and the sequencing is not targeted, and it sequences all the genetic material in the community. And then lastly, we have metatranscriptomics, which is the most expensive, and it assesses what are these microbes doing. And it's best when it's coupled with shotgun metagenomics because you can use those de novo genomes from the shotgun metagenomics as reference genomes. And then you can align your reads to them, which is the raw data, and it allows one to model the activity of certain genes or pathways. And this is really useful for time series studies. So NewAtlantis Labs uses these technologies together to achieve different conservation goals. So how can we streamline metaomics to accelerate discovery and conservation? Well, each step requires several software packages, and many of these packages are difficult to install. A lot of them are incompatible with each other, and most of them require large structured databases. And so it requires a certain expertise for quote unquote best practice analysis, and that's knowing which tools to use, how to use them, their assumptions, and it also requires intermediate data reformatting. So a lot of the outputs, they don't work perfectly with the inputs required by other tools, so you need to do some reformatting. And a lot of these tools require the same types of intermediate data, so you can minimize redundant computation by reusing these intermediate results. So sometimes that requires getting like the actual package and then forking it and then making some modifications so it can work better with our system so we can optimize those resources and also decrease costs. And also understanding how to allocate the resources properly, which is often overlooked. So if you over provision, then you're going to be paying for resources not being used, so that's wasted money. And then if you under provision, then your system crashes and you have to rerun it with more resources, so that's kind of like twice the cost. So there's a lot of cost saving in knowing how to allocate resources effectively. The NewAtlantis Labs ecosystem is powered by PISCES, which is the Precision Intelligence for Sustainable Computational Ecosystem Science. And this is our marine AI platform for optimized compute, agent-based workflow orchestration with natural language support, and data storage. And this allows us to do reproducible science. And we develop a lot of open source peer-reviewed software. So you can see our flow chart below. On the left, we have our data ingestion pipeline. So we're getting data from public resources like NCBI or ENA. We also have research labs. So this is like if somebody gets a grant and then they get sequencing, they can send us a sequencing. And then there's also organizations like MPAs. And then we have our different omics technologies, and then that feeds into our PISCES platform. So there's a couple different toolkits right now. And once we have these contextualized genomic assets, what we do is we put them into our knowledge graph in our genomics registry and our model registry, and then we can use them for conservation insights. So like biomonitoring, bioprospecting, and forecasting, and this leads to marine conservation. And there's a subset of these tools in purple, which is called microcosm analytics. And this is our microbiome analysis toolkit that runs on the PISCES platform. And Jay will give you a demo on this later. So one of the tools in the toolkit is Viva, which is an open source software suite that we use. It's modular and it's for end-to-end metagenomics and metatranscriptomics analysis. We also have Sedna, which is our bioprospecting and biosynthetic gene cluster detection algorithm. We have Leviathan, which is our taxonomic and functional profiling toolkit. And then we have niche space, which is for calculating metabolic niche space and dimensionality reduction. And we also have CGEM Forge, which is what we use for community scale metabolic models. So I mentioned knowledge graphs a couple of times. A knowledge graph is a next generation contextualized database, and this contextualizes the genomic assets for intuitive data exploration, and it connects the genomic assets in a biologically relevant way so we can query the database with natural language to produce a contextualized response instead of the LLM just hallucinating and then generating what it thinks sounds correct but isn't actually correct. So this allows us to have a conversation with our data and extract deep biological and ecological insight. In our LLMs, they use the graph ontology when computing the response. So you can see our ontology. We have samples that feed into genomes, and within genomes, we have BGCs and proteins. And then some of those proteins are enzymes, so they catalyze reactions, and those reactions produce compounds. And you can see on the right is our platform. So we're asking a query. It's giving us a summary. It's showing us the cipher query in the middle that it generates. And then we have all of the connections that are relevant to that query. Microcosm analytics streamlines research to unlock value. So we found a lot of power in analyzing new data sets, but also reanalyzing legacy data sets with microcosm analytics. So most studies ignore eukaryotic organisms like protists and fungi, and also viral genomes. So microcosm analytics is designed specifically to handle all domains of microbial life. So this is everything from fungi and diatoms all the way up to bacteria and archaea and phages. So it can handle all of these. And our genome recovery workflows typically recover between 2x and 8x more high quality prokaryotic genomes. And this is largely because of the state-of-the-art binning algorithms we use. So we're constantly looking at PubMed, looking for new binning algorithms that come out, and we find one that's appropriate, then we'll incorporate it into our pipeline. And sometimes that involves us forking it and having to make some modifications so it can work a little bit better and be more streamlined. And we also have an iterative binning procedure. So we have a bunch of different binning algorithms we use. We bin individual genomes from the metagenomic assembly, and then we dereplicate them, and all of the context of like those large contiguous sequences that we get from the genome assembly, the ones that don't pass QC get fed back into the binning algorithm. So we're really able to squeeze like more juice out of the lemon, and all the other tools that we know of at this moment, they only do one round of binning, so they're often throwing away a lot of data. And we've found new genomes in iteration 2, iteration 5, 10, all the way up to 20, depending on how deeply sequenced the data is. And most other pipelines would just toss these out. So microcosm analytics contextualizes genomic assets. So we know which gene comes from which genome, we know which genome comes from which sample, and most importantly, we know the environmental conditions associated with each sample. So we know the lat, ln, depth, pH, temperature, all that information that's recorded. And this is essential for building a contextualized knowledge graph. And environmentally contextualized genomic assets are really the key to unlocking the value with metaomics. So microcosm analytics, it really just makes metaomics accessible. So you can run workflows without the pain of setup and deployment. So you don't have to install the softwares, compile the databases, provision resources, and you also have access to microcosm analytics dashboard. So these data apps that you can use for exploration, and they're easy to understand interactive reports for insight, which is accessible for general users and power users. So think like conservationists, data scientists, policymakers, you name it. Microcosm analytics accommodates a wide range of user objectives. So this is really useful for research institutions that have a grant and they're trying to do hypothesis testing, biotechnology and pharmaceutical companies that are interested in bioprospecting for novel biomedical compounds or industrial enzymes, and conservation organizations that are interested in ecological health assessment. So I'm going to shift it over to Jay, our chief scientist, for the microcosm analytics demo.
Thanks, Josh. Can you see my screen now?
Yeah.
Perfect. Good morning, everyone. I'm excited to introduce you to Microcosm Analytics, our Metaomics Toolkit. This screen here is our interactive dashboard, which is the user interface that makes complex data come to life in a visual and intuitive way. And it's important to understand that while the dashboard provides a user-friendly experience, in the backend, we have all the sophisticated systems that do all the heavy lifting in terms of data processing, analysis, and database query. So Microcosm Analytics offers a powerful way to understand the invisible world of microbial communities, transforming simple water samples into valuable insights for conservation and sustainable development. Conventional monitoring initiatives typically result in large taxonomic inventories, but lack the ability to characterize biological activities or interactions among organisms that are the main drivers of ecosystem functions. However, with microcosm analytics, we are able to delve deeper into this complex microbial dynamics. The spider chart shown here illustrates our competitive edge in microbiome intelligence across 8 critical capabilities. And preliminary market research indicates that we can outperform the industry average. The green line in this chart represents substantial advantages, especially in multi-kingdom analysis and biosynthetic discovery. And what this means is very simple. While traditional monitoring tells you which microbes are present, we can characterize what they are actually doing. So we are tapping into the 99% of ocean biomass that's functionally unknown. And every spike on this chart translates into deeper insights, including early warning signals, as well as opportunities for valuable discoveries to support conservation efforts. Now let's concentrate on real samples collected from marine environments. So here's a single sampling campaign conducted by our colleagues at the Smithsonian Tropical Research Institute in Panama, where they collected samples from mangroves and coral reefs across the Atlantic and Pacific Ocean basins. Our extensive suite of analytical tools enables deep understanding of microbial community organization across diverse habitats and ocean basins. So now let's focus on these patterns and paying particular attention to the beta diversity analysis. Its distinct communities in the Pacific and Atlantic Ocean basins, effectively distinguishing between mangrove and reef microbial communities. And each color signifies a unique microbial signature. And these findings reveal distinct ecological zones and illustrate how environmental factors influence the structure of biological communities. Our extensive suite of analytical tools also enable us to identify the functional potential of microbial communities. And this holds significant value in biodiversity accounting terms. To illustrate this, let us concentrate on this map of biosynthetic gene clusters, which are genetic blueprints for producing high-value chemical compounds with potential industrial applications. Across all the samples analyzed, we discovered approximately 600 unique biosynthetic gene clusters, and our analysis demonstrate that the relative distribution of different chemical classes of biosynthetic gene clusters, including the dominant polyketide and terpene classes, exhibits significant variations between mangroves and coral reefs. So what this findings suggests is that evolution has optimized chemical profiles of the marine microbiome to suit the specific conditions of each environment. But also notice here the red rectangles. Many of those correspond to unclassified biosynthetic gene clusters, and each of these could be tomorrow's breakthrough. For instance, a new antibiotic, a plastic-eating enzyme, or even a more efficient carbon capturing metabolic pathway. And we know exactly where to find them and under what conditions they thrive. So this microbiome intelligence, accessible through microcosm analytics, presents significant potential to help eventually transform marine protected areas into centers of innovation capable of independently financing their conservation efforts. The snapshot dataset from Panama provides valuable insights into the state of the marine environment at a given point in time, showing us what is present in their waters. However, these snapshot samples offer only a partial picture of marine ecosystems, much like a single weather observation offers a glimpse, but not a climate forecast. These snapshots alone are insufficient for understanding long-term ecological dynamics. Therefore, integrating time series datasets collected through continuous monitoring is critical in order to develop robust ecological forecasting model prototypes, despite the numerous intrinsic difficulties and possible weaknesses associated with time series analysis. And we are currently analyzing a model in a few publicly available marine metagenomics time series. And in this demo, we will focus on time series bloom forming events, where the same site has been monitored for several months. And here, I would like to emphasize that the purpose of this is to just illustrate the possibilities of time series data to observe changes over time and gain deeper insights into ecological processes. So in this chart, observe the blue trajectory, which tracks the species richness over time. Here we can see substantial drops at the specific time points where the community seems to experience difficulties in adapting to random signals or true environmental drivers. But here's the critical insight. Substantial declines in the species richness may often precede significant alterations in water quality or marine ecosystem health. And we can use other classical metrics in order to keep track of changes in the community, such as Shannon and Simpson diversity indices. It is also important to understand the pace of ecosystem change, not just changes that are occurring. So this chart here illustrates ecosystem turnover, which with each spike representing points where the microbial community experiences significant shifts. So in a nutshell, we compute this by assessing beta diversity between subsequent time points, and the orange triangles here in this time series can highlight critical transition events that exceed a predefined threshold. And inspired by simple but well-defined statistical approaches like this, the ultimate goal is to identify ecosystem tipping points impacting the entire food web, days or weeks in advance. And it should be noted again that these insights come from preliminary analysis and serve solely for illustrative purposes. The next chart here illustrates one of our early prototypes for developing predictive warnings in marine ecosystems. The orange line, the orange line in this chart represents a preliminary warning index that we're prototyping. And the red triangles mark instances where our prototype potentially detected risk signals prior to observed disruptions. And we are actively exploring pattern recognition, anomaly detection techniques, as well as causal inference methods to examine subtle changes that might precede major events. And as we gather more data across diverse locations and conditions, we hope to improve the reliability of these indicators. So this is the promising vision. Imagine having weeks of advance notice for events like harmful algae blooms, or vulnerable spawning periods, or declining fish stocks. While still under development and requiring experimental validation, these approaches hold the potential to transform ocean management from reactive responses to proactive prevention. And based on an extensive review of the scientific literature, which guides our current prototype development for early warning systems, here we want to briefly present an initial comparative analysis that emphasizes certain benefits of time series datasets relative to a snapshot sampling. For instance, this exercise suggests that continuous monitoring may provide up to 6 times better early detection capabilities compared to traditional snapshot sampling. In addition, early results seem to indicate potential increases in prediction accuracy by approximately 7-fold and possible improvements in risk reduction by around 5 times. So overall, this preliminary analysis highlights a potentially significant shift in ocean conservation, moving from reactive management to proactive predictive strategies. So with these promising insights into the advantages of continuous monitoring, it's important to emphasize that our biodiversity tracking is simpler than you might expect and requires minimal infrastructure. For instance, for basic bioprospecting, simply hand scoop sediment, freeze it, and ship it to the designated lab. For broader biodiversity surveys, filter seawater, preserve the filters, and send them to the appointed genetics labs. And we are here ready to support or help build local capacity for both field sampling and laboratory work, including DNA sequencing. And we can design flexible toolkits tailored to your objectives, budget, and infrastructure. And from experimental design to visualization to data visualization, we can handle the technical complexity, delivering intuitive dashboards that highlight key spatiotemporal patterns and ecological drivers. So this potential for more effective and efficient monitoring, as highlighted in this dashboard, led us to a key point. For the first time, we have the possibility of truly understanding the ocean's living engine, and that is the complex microbial network that sustains all marine life. And this understanding can translate directly into better conservation, whether protecting coral reefs, discovering new species, or developing innovative financing mechanisms. Microbiome intelligence provides insights we have never had access to before. And one of our goals is to build a global network of partners who share this vision. Partners who understand that the smallest organisms are likely to hold the biggest solutions. So with this, I'll hand it over to my colleague, Florence. I think you are on mute, Florence.
Am I sharing the right screen this time?
Great. Okay, so where are we today? You have seen that the team has developed the PISCES platform and several tools with the demo that Jay just did of the microcosm analytics as one of the application. And all of those are at the functional prototype stage, and we already piloting this with a research institution. The feedback has been incredibly encouraging, and looking ahead, we are preparing for wider platform launches, new partnership, and also funding opportunities to build pilot sites and MPA use case, and also to continue improving our technology, especially around the data integration for user experience. So we believe that for funders, researchers, decision maker, conservation practitioner, and also investor, NewAtlantis can represent a unique opportunity to engage and transform ocean stewardship. So let's talk about how we are building value and impact through potential partnership and collaboration. So we can start anywhere on this wheel. I will start with the scientific collaboration, because that's really something very important to work with a diverse network of national labs, academics, universities, open science contributors, and also conservation groups. The goal here is to advance cutting-edge metagenomics research, as you have seen, validate our ecological insights, and also generate discoveries that can lead to new patents and real-world solutions. These are really core. We want to turn raw data into high-impact science and even new product that can be really used. Then we have technical alliance. So those strategic partnership can be with tech accelerators and open platform. We aim at keeping PISCES, the global platform, integrated, standardized, and scalable, while expanding its reach beyond genomics. So we can integrate physical chemical sensor streams, satellite and drone remote sensing layers with other biomonitoring data sets, and also do that with automated sampler that are needed for the time series collection. So if we continue to embrace truly transdisciplinary solution, we can unlock powerful synergy that can transform all this disparate data into coherent decision-making marine intelligence. We're looking also for impact-focused funding partnership, because it's really key. Here we can we're looking at blended finance capital, public-private partnership, investors, impact investors, philanthropy, and also multilateral development banks. And this mix of funding could accelerate the global rollout of the platform itself, build its core data sets by collecting the raw biological data directly from the ecosystems and particularly the MPAs. And specifically, it could fund projects that could enable to build time series sequencing. And those are really key, as Jay have already mentioned, because high quality marine time series omics data are really scarce, and yet they are really essential to create the baseline biodiversity, long-term monitoring, that are really comprehensive and scalable for conservation planning, but also for adaptive management. Also to build accurate modeling, and for the creation over time of digital twins, making these data sets uniquely valuable for risk assessment, mitigation, but also for bioremediation strategies. And it will enable as well to build the foundation for ecosystem services quantification, which can form the foundation for valuing metrics or valuation metrics for financial products such as carbon and biodiversity credits. With the governments, we are working closely with some of them already, and governmental agencies, MPA manager, indigenous local communities, and of course, conservation partners. By integrating PISCES insights into national marine conservation strategies, and for example, marine spatial planning, we can help build local capacity, improve data infrastructure, and unlock both economic and social benefits in those countries, all within a framework that is compliant with access and benefit sharing rules implied by the Nagoya Protocol. And finally, our commercial partnerships, so engaging with pharma, biotech, cosmetics, industry, insurance, and bioremediation. These partners can access to premium PISCES data and analytics, opening up new opportunities for breakthrough products and exclusive markets. And so in a nutshell, all partnerships are designed to create a win-win, advancing science, supporting conservation, and unlocking real economic value for everyone involved, from local communities to global industries. So if you want to connect, you can contact us for any queries or demo that is really tailored to your needs, either through our website or the address that you see there. And for those who will be in Nice, Josh will be presenting a poster session at the Ocean Science Congress, and you can find us there, and then at the Blue Economy Finance Forum in Monaco, and also at UNOC in Nice. Thank you very much.
Thank you, Florence. Thank you, Josh and Jay. I see some people uploading here. Yeah, your presentation sounds like what you're building is coming from the future, but it's much needed today and ASAP. So a couple of notes. First, this webinar is being recorded. You will get a notification through Luma when the recording is going to be live. Usually takes us about a week or two. We try to summarize the webinar in a blog post as well, so you get those notes and you get the full recording. And now moving to questions. I've seen a few questions that Christian has been addressing already. One was on type of roles and skills in the ocean space. All skills are welcome, and Christian has a bit more detailed response there. There was another question about the legal frameworks for this to happen in terms of, access to data sharing. And Christian has elaborated as well on that. Luisa asked about fresh water, whether you know, metagenomics could be used as well for fresh water, and the answer is yes. One other question that has come from Sriram, he is asking, in order to get all of this data for a specific geographic location, would that not be immensely expensive? Or is the goal to reduce the cost of data collection and accuracy of analysis even with less data? I heard during the presentation that the goal is actually the latter, to reduce the cost. Who wants to take that? Is that you, Florence, or Josh?
I'll take that one. So the cost of sequencing is decreasing rapidly year by year, and there's new sampling techniques, so we can use different platforms so we can get more data with less costs. So the idea is just to collect the baseline now, and I think getting a time series is really important for that. And in the future, I mean, as sequencing costs go down, like this whole process will be a lot cheaper and be more affordable.
Sriram, I'll unmute you. I see you're raising your hand. You could also do it with Zoom. Go ahead.
Perhaps, sorry, if I may add to that answer, because the costs are being reduced on one hand, but also what is interesting is that what we are promoting is the idea that an MPA, for example, may engage in this type of biomonitoring and not only gain like a very deep understanding of the ecosystem, which is obviously useful for management and so on, but it's also possible to get these, let's say, side products in the form of DNA sequences that code for useful protein, can be an enzyme that can be useful for making pharmaceutical products, for example, and that can generate a revenue stream for the marine protected area. So you can have money to continue biomonitoring, continue the understanding the ecosystem, and protect it in the long run. So that's why NewAtlantis is so interested in generating this revenue stream, so that protection can be long lasting and not only last for 4 years or 2 years, what is typically the span of a scientific grant, for example, which is what we have so far.
So is that the to my understanding, these marine protected areas are going to be under usually governmental or like some kind of public ownership. And you mentioned in this, I assume what you're referring to is the intellectual property that you refer to in the written comment, or the written answer is the same thing that you're referring to with these sequences that could be potentially used for pharmaceuticals. Is there a conflict there? Would that end up are there legal frameworks by which you can take something from this public resource and then basically copyright it in some way?
Florence?
Go ahead. I know you.
All right. I think Florence is the person to answer, but I would say what I was saying in the comments over there is like, by the way, your question is great. I mean, it's really central to this. I mean, it's really, really important, right? So we're talking about how do we do to engage in this type of commercialization of genetic or marine genetic resources, as they are called, in a fair way, so that the profits also go down to where it was found, that the DNA sequence in the first place. So right now there are mechanisms and legislations in place that make this a lot easier than before. Foremost is the Nagoya Protocol under the CBD, and there's also another new one for international waters being developed. I don't think it's implemented yet. So there is legislation and the mechanisms are already in place. And from there, I would pass the floor to Florence to elaborate more.
Yes, so there are two things. One is actually the international legislation that, of course, we will be compliant with. So this is the access and benefit sharing framework that are under the Nagoya Protocol, and countries are normally developing those at different stage. Let's say that some countries have already all of the ABS system in place, and some others are still have to do this. But we will be kind of preempting all of this in how we develop and use those marine genetic resources. And Jay can talk more about the data ownership when it comes more precisely to this. But if I talk into the benefit sharing mechanism, what we envisage is that the revenues that are generating through the commercial products that will be creating along the line, this is really benefit sharing with the government, with the local communities, or the different partners that are involved. Let's say if this happened in a marine protected area, we would call those the stakeholders of this marine protected area. So depending on who's actually funding some of the costs and the revenue that are generated, this will be discussed with a transparent view over the cost. But then the benefit sharing should at least 60% of those revenues generated by the government should be reinvested into the marine protected area or into the conservation actions. This is how we can ensure that over time, this virtuous cycle is created by generating new products, but investing the revenue into conservation. So maybe, Jay, you want to add more specifically how we can ensure the ownership and the origin of the data?
Yeah, definitely. We are Nagoya Protocol compliant for sure, and there are some technological advancements that enable full transparency in the benefit sharing, such as blockchain technology, for instance, where we can actually turn all the rules necessary for the benefit sharing. I think you are mute, Florence.
Yeah, I was just saying that you went off. So does that answer your question?
Yeah, that answers my question. Thank you so much. That was a really detailed answer.
Perfect. I don't see any other questions so far. Oh, there's one on the use of blockchain. But before moving to that, I see some people in the audience who are involved in the blue carbon space and also coral restoration. I was wondering, because I've heard the coral, I heard carbon capture in the presentation. Are there any, what are the applications for blue carbon developers, for coral restoration projects of this of metagenomics and your platform in particular?
Jay?
Yes, on that note, so Metagenomics offers an intriguing possibility to enhance carbon valuation frameworks, for instance, by examining the microbial processes that underpin marine carbon cycling. That's one of the additional blue carbon assessments primarily focus on physical carbon stocks in sediments and biomass. But emerging research suggests that microbial communities can actually play a fundamental role in carbon transformation, sequestration, and stability. So through functional analysis, including gene and metabolic pathway analysis, We can potentially identify and quantify microbial processes like carbon fixation, methane production, and organic matter degradation that influence whether carbon remains stored or returns to the atmosphere. I don't know if that answered your question.
On blue carbon, yeah, totally. And what about coral restoration?
Josh, you want to take this one? You're muted, man.
Sorry about that. So for coral restoration, we haven't had any projects specifically for that, but there were some projects in the work, and we're working previously on probiotics for different corals. So in previous research, what we've done, or we've collaborated with researchers that have gotten microbial communities for different corals, and then they had some beneficial microbes that they've added to these communities, and then they were able to like increase the temperature in certain communities and have these microbes actually make them more resilient to these increase in temperature. So that's just some research that we're building on, but this is definitely an area of active research. And coral microbiomes are notoriously difficult to study because their endosymbionts have very, very large, complicated genomes from like tertiary endosymbiosis. So there's a lot of dinoflagellates in there, and they're quite complicated because we don't have the actual genome sequences for a lot of these corals. So when we do metagenomic sequencing, it's hard to differentiate what's the host genome and what's the microbial genome. So we're kind of looking at it as a whole community together, but they're very, very difficult to study.
Great, fantastic. There was a follow-up on blue carbon. VJ is asking, can this approach be used in green carbon as well? So in terrestrial ecosystems.
So I was responding to that in the chat, but yeah, there's definitely companies out there that engineer like microbial consortia that they would put in soil to like increase like water retention and improve crop yield, and also using in like forests and stuff. But yeah, those companies are out there, and luckily these tools that we're developing, I mean, we're borrowing from geology, we're borrowing from like human microbiome research, like the science itself is very collaborative, so we can learn a lot from all the different domains. And as a developer, we really emphasize developing tools that are generalizable. So even like that niche space package and like Leviathan, like all of our toolkits, like they can work for green economies as well, and also for human microbiomes. So we're just trying to push science forward in general, not just for marine science, but we're focusing on marine science.
Great. I see Christian has answered the question on the use of blockchain. There's another one from Sriram on why have past studies neglected microbial viral genomes. Maybe it was in neglect, maybe it was not the capabilities were not there. Josh, you want to take
Yeah,
that?
so at least with eukaryotic genomes, eukaryotic genes are so first off, the genomes are very complicated. Like there can be multiple chromosomes in there. There can be different duplicates, events. Also the genes, they have these things called exons. So like with the bacterial or archaeal gene, those are prokaryotes. The gene is just read in like one nice section, but with eukaryotes, it's broken up into sections, and sometimes those intermediate parts are like very, very long. So the gene prediction software is complicated to run. And usually when people are doing eukaryotic gene prediction, they're doing it for like model organisms. So if you're doing it for like humans, or if you're doing it for like mice or like or certain crops, like those are model organisms. So there's specific algorithms that work for it, but not until recently have people developed like a generalizable approach that we can use for metagenomics, where we don't actually know the taxonomy for the organism. And also the taxonomy for both eukaryotes and viruses are really complicated. I mean, viruses, they kind of border on what is life. Like some people say they're live. I think the consensus is still that they're not, but then there's also like megaviruses that have their own tRNA. So it's very complicated, and viruses are just, to be honest, they're just weird. So a lot of people have just ignored them. But there's been a lot more tools recently that have allowed us to like confidently pull out viruses and also assess their completeness. So the reason why this is possible now is because we have algorithms that can compute the genes and recover the genomes. We can compute the quality of the genomes, and also the taxonomy of the genome. So those are all recent developments, and microcosm analytics is incorporating those newest tools. And like I said, we're constantly looking for new tools to make our methods better.
Fantastic. We're at the top of the hour, but I had a really question on this. Like you mentioned the Panama example. Are there any other real MPA conservation management examples where metagenomics have been really helpful so far?
Christian, can I answer this? I think you're muted, Christian.
Sorry, I am being bombarded by messages here. I did not hear the question.
No worries, I'll repeat it. What are some other real-world examples where metagenomics have been contributing to marine conservation and MPA management, besides the Panama example that Jay walked us through?
Okay, so I would say this is an emerging discipline, and whatever paper we find about applications are fairly new. And we've seen applications in coral reef restoration. I don't know if Jay already mentioned that. Jay, did you? So in coral reef,
Josh answered that.
yeah. Okay, so that is one. Another one is in aquaculture industry. It is used to understand both what is going on within the enclosures, for example, with the internal microbiome and associations of the target species with non-target species that coexist with them. It could be symbionts or parasite fauna, for example, but also outside of the cultures where you have the surrounding biota that is being impacted by highly dense packed salmon, for example, in the south of Chile, or other places. And what else? There are not many examples around functions of the ecosystem functions, and how they are being disrupted by very different types of stressors, such as pollution or overfishing. And those are things that we are looking forward to find out. But it's certainly, we will find signals, because ecosystems are a really intricate network of connections. Some are positive, some are negative. If you alter some of those nodes, which especially those that are key, that are central in the networks, the overall network, responds in sometimes unpredictable ways, which we think we can make them predictable by understanding the functioning. So we are betting at understanding the nuts and bolts that goes within complex network, and be able to make predictions about what would be the scenario if we continue putting some type of stressor, or what would happen if we try to do some remediation. I don't know if that let's answer the question.
Yeah, fantastic. We'll take one last one for another minute, and then we will close this webinar, and I really appreciate your time. So Rafael is asking, I think it has been answered about the safeguards in place to avoid that work or the platform and data getting used for nefarious purposes. I guess Josh responded to that. So I just want to echo what I've seen in the chat. Congratulations on what you've built so far. And all the best on growing this platform and putting it in the hands of MPAs, governments, local communities who need it. Really looking forward to your presentations at UNOC. And yeah, thank you so much. Everyone, you will receive a notification on Luma when the recording is available, and I'll put the contact of the NewAtlantis Labs team in the chat. So feel free to reach out to them. Thank you everyone for joining today.
Thank you very much.
everyone.
Bye.
Bye.